Nearly a kilometer beneath the surface of the Gulf of Mexico, something is still alive. It has been alive since steam engines reshaped the continents above, since electricity first reached the dark, since photographs were rare and the modern world was only beginning to form. It has never eaten, and yet it continues to grow. Pressure bears down with the weight of mountains. Hydrogen sulfide seeps from the seafloor, a gas lethal to most animals. No sunlight has ever touched this depth, 400 to 900 meters below the waves, where cold seeps leak methane and chemicals into water that remains stable for centuries. Rooted upright in sediment, it rises millimeter by millimeter through decades that pass unnoticed at the surface, an animal that should not be alive at all. There are individuals alive right now that were already living when Abraham Lincoln was a child. Growth studies suggest some can endure for more than 200 years. They were born with a mouth, a simple digestive tract, the basic tools of animal life. Then it vanishes, the mouth disappears, the gut dissolves permanently. They do not hunt, they do not graze, they do not swallow or filter feed. Instead, a red plume extends from a pale tube, its color shaped by hemoglobin that binds hydrogen sulfide from the surrounding water. Inside their bodies, billions of bacteria convert those toxic chemicals into energy through chemosynthesis. No sunlight, no photosynthesis, food built from chemicals rising through the earth itself. Scientists first encountered these dense communities in the late 20th century, descending in submersibles and finding ecosystems thriving where none were expected. Entire worlds powered not by light, but by chemistry. But how can an animal survive for centuries without ever eating? What kind of life depends not on sunlight, but on chemicals seeping from the sea floor? How does a creature lose its mouth and begin to thrive instead of starve? And what does this mean for the definition of food itself? Tonight, we will drift slowly down toward the cold seeps and stand beside these towering worms as they continue their slow growth in total darkness. But before we begin, if you enjoy calm and soothing explorations like this one, filled with wonder, science, and the gentle unraveling of life's mysteries, I'd love it if you gave this video a gentle like and maybe tapped subscribe. It helps more curious minds find their way to this peaceful little corner of the internet. And if you'd be so kind, let me know in the comments, where are you winding down from tonight and what time is it there? It's always a joy to see how far across the world this little sleepy corner of science travels. Now take a deep breath, let your shoulders soften, and let's begin. Far below the restless surface of the Gulf of Mexico, the seafloor holds quiet fissures that never close. From these openings, invisible streams rise in slow motion, not of heat or flame, but of chemicals drawn from ancient reservoirs buried deep within the earth. These places are known as cold seeps. They do not erupt or roar, they breathe. At depths ranging from 400 to 900 meters, sunlight has long since dissolved into blue and then into nothing at all. What remains is a steady twilight that never brightens and never fades. The water is cold, hovering just above freezing, and the weight of the ocean presses downward with immense force. Each square inch bears pressure dozens of times greater than at the surface, a constant embrace that shapes every body adapted to endure it. A cold seep begins far beneath the sediment, where organic material buried over millions of years has transformed into methane and other hydrocarbons. Tectonic stresses and subtle fractures allow these gases to migrate upward. Along with them comes hydrogen sulfide, produced by microbes that feed on buried compounds in the absence of oxygen. The chemicals move slowly through layers of mud and sand until they reach the ocean floor, where they seep into the surrounding water. Unlike hydrothermal vents, these sites are not defined by heat. The temperature remains nearly the same as the surrounding sea. What distinguishes them is chemistry. Methane drifts upward in shimmering threads. Hydrogen sulfide dissolves into the dark water, toxic to many forms of life, yet rich with potential energy. The seep may appear still, but it is chemically alive, a boundary where geology meets biology in continuous exchange. The depth ensures permanence. Storms that churn the surface never reach this far. Waves do not disturb the sediment. Seasons pass unnoticed. Even climate shifts unfold slowly, filtered through layers of ocean that buffer sudden change. Beneath this heavy water, the environment alters on geological timescales rather than human ones. Pressure at these depths compresses gases and stabilizes structures that would collapse nearer the air. Methane hydrates, crystalline cages of water and gas, can persist within the sediment for centuries. As long as the subsurface reservoirs remain intact and the fractures stay open, the seep continues its patient release. Flow rates may fluctuate slightly, but the source endures. In this darkness, stability is not a brief condition, but a defining feature. The chemical gradients remain steady year after year. Oxygen drifts down from distant surface waters through slow global circulation, meeting sulfide in delicate balance. The seafloor here is not barren rock, but a chemically sustained landscape, persistent and unhurried. Within this measured world, life does not depend on the rising sun. It depends on the quiet constancy of the earth beneath, and on the slow exhalation of gases that have waited millions of years for release. Morning never arrives here. There is no horizon to brighten, no gradual lifting of shadow into color. In this depth, the idea of daylight belongs to another realm entirely, yet life persists, not as a faint trace, but as a structured and enduring presence. On the surface of the planet, most living systems lean toward the sun. Plants gather photons and weave them into sugars through photosynthesis, forming the base of food webs that ripple outward into forests, grasslands, and reefs. In the deep sea, that luminous foundation is absent. No leaf unfurls, no algae drift in green clouds. The chemistry of survival must begin elsewhere. At cold seeps, energy rises from below rather than falling from above. Methane and hydrogen sulfide seep from the sediment, carrying within their molecular bonds stored potential. Certain bacteria possess the enzymatic tools to unlock that potential. Through chemosynthesis, they oxidize these chemicals, drawing electrons from sulfide or methane and transferring them through internal pathways that resemble delicate electrical circuits. The released energy allows them to bind carbon dioxide into organic molecules, forming sugars and other compounds that sustain growth. The process unfolds silently within microbial cells, invisible yet transformative. Where sunlight would normally drive carbon fixation, chemical reactions now assume that role. Oxygen, drifting down from distant surface waters, becomes a partner in the exchange, accepting electrons and enabling the chain of reactions to continue. In this way, a gradient of chemistry replaces a beam of light. These microbes do not exist as isolated specks. They gather in mats across the sediment, forming pale fields that ripple faintly in slow currents. Others live within the tissues of animals, converting dissolved chemicals into nourishment in exchange for shelter and stability. The seep becomes a site of continuous conversion, where inorganic compounds are steadily reshaped into living matter. Because the chemical supply is persistent, the productivity can be steady as well. The energy flux is modest compared to a sunlit meadow, yet it is reliable. Carbon fixed by bacteria enters surrounding bodies, supporting worms, clams, mussels, and crustaceans adapted to this chemistry. Each organism is linked, directly or indirectly to the initial microbial alchemy occurring at the boundary between sediment and sea. In this world, food webs do not taper upward from plants, but from microbes that harvest energy from the earth itself. The absence of light does not signify absence of life. Instead, it reveals an alternative architecture, one built on oxidation rather than illumination. Here, chemistry becomes currency, molecules replace rays, and from the slow exchange of electrons in darkness, an entire ecosystem takes shape, sustained not by the turning of the planet toward the sun, but by the steady release of compounds from within it. The first clues did not come from sight, but from instruments lowered into depths few humans had ever witnessed. In the latter half of the 20th century, research vessels traced irregular chemical signatures along the Gulf seafloor, readings that suggested something unusual was occurring far below the reach of nets and dredges. Methane concentrations were elevated. Sulfide levels hinted at active processes beneath the sediment. The data felt incomplete, like a map with a blank space at its center. When submersibles finally descended to investigate, their lights cut narrow corridors through the surrounding dark. What appeared in their beams unsettled long-held assumptions. Instead of a sparse landscape dotted with occasional scavengers, the seafloor supported dense clusters of life. Pale mussels pressed together in thick beds. Long white tubes rose upright from the sediment in quiet forests. Crabs moved deliberately among them. The scene possessed structure and abundance where theory had predicted scarcity. For decades, marine biology had been guided by a simple expectation. The deep ocean was thought to depend almost entirely on organic matter drifting down from surface waters, a thin rain of detritus known as marine snow. In that framework, productivity declined steadily with depth. The discoveries at cold seeps suggested another source of energy entirely, one rooted in chemistry rather than sunlight. The early dives were cautious and deliberate. Pilots maneuvered mechanical arms to collect samples, aware that each fragment retrieved from the bottom might revise textbooks. Cameras recorded unfamiliar species, many lacking obvious ties to known shallow water relatives. Specimens brought to the surface revealed tissues infused with bacteria, anatomical features adapted not for grazing or predation, but for partnership with microbes. These explorations unfolded alongside similar revelations at hydrothermal vents in the Pacific. Together, they redrew the boundaries of where complex ecosystems could exist. The deep sea was no longer a passive recipient of surface productivity. It contained autonomous systems, fueled from within the earth itself. Entire communities could assemble around chemical gradients, sustained by microbial metabolism and the organisms that depended on it. The impact extended beyond taxonomy. Biogeochemical cycles were reconsidered. Models of carbon flow expanded to include pathways independent of photosynthesis. Evolutionary narratives incorporated lineages shaped by isolation and chemical adaptation. Even the search for life beyond Earth absorbed the implications, recognizing that sunlight need not be the sole architect of biology. In the quiet footage from those early submersible dives, one can see the careful sweep of artificial light across clustered shells and rising tubes. The beams move slowly, as if aware they are illuminating more than a patch of seafloor. They reveal a principle that had waited unnoticed beneath layers of water and sediment, patient and intact until human curiosity reached down and found it. A slender column rises from the sediment, pale and upright, as though the seafloor itself had grown a quiet spine. This is not the animal's body in the usual sense, but a structure it builds around itself, a tube secreted gradually over years. Layer by layer, The worm releases chitin and proteins that harden into a flexible casing. The tube anchors deep within the substrate, its lower end buried securely, its upper length extending into the surrounding water. It is both shelter and scaffold, a lifelong dwelling that lengthens as the animal within slowly grows. The outer surface is smooth, sometimes faintly ridged, resistant to abrasion and intrusion. Small organisms may settle upon it, but few penetrate it. The tube shields the soft tissues inside from scavengers and from direct exposure to shifting sediments. It absorbs the steady compression of the surrounding water, distributing pressure along its length. Within this narrow cylinder, the worm's body remains hidden, protected from view and from harm. Inside, there is no rigid skeleton, no segmented armor. The worm itself is delicate, pale, and elongated, its tissues arranged in a series of regions specialized for exchange and support. Muscles line its inner wall, allowing it to retract deeper into the tube when disturbed. A central cavity carries fluids rich in dissolved gases and nutrients, circulating slowly through vessels that branch and reconnect like fine threads. At the upper end, emerging from the safety of the tube, a plume unfurls. It spreads outward in soft filaments, forming a structure that resembles a cluster of petals suspended in water. This plume is richly supplied with blood vessels, each filament increasing the surface area available for exchange. Through these tissues, the worm absorbs oxygen and other dissolved compounds essential to its internal partnerships. The plume's color is a deep crimson, striking even in the filtered glow of submersible lights. That color comes from hemoglobin, a molecule also familiar in human blood, yet here it carries an additional role. This hemoglobin binds not only oxygen but hydrogen sulfide as well, holding the toxic molecule in a controlled embrace. In most animals, sulfide would inhibit cellular respiration, interfering with enzymes that depend on oxygen. In this worm, specialized hemoglobin prevents such disruption, transporting sulfide safely through the bloodstream to the tissues where symbiotic bacteria reside. The circulatory system functions as a quiet courier service, delivering both oxygen and sulfide to internal regions dedicated to chemical conversion. The plume gathers these ingredients from the surrounding water, then retracts or extends subtly as conditions shift. It is the only visible sign of the animal's living presence above its tube. Encased, supported, and supplied through this architecture, the worm's body plan reflects a life rooted in place. It does not wander. It builds upward, enclosing itself in protection while opening a single red crown to the water, balancing exposure and shelter with patient precision. Before it becomes a rooted presence in the sediment, the worm begins as something almost weightless. Released into the open water as a microscopic larva, it drifts within slow currents, carried across distances its later body will never travel. At this stage, it is small, translucent, and complete in a way its adult form will not be. It possesses a mouth. It possesses a gut. Cilia beat along its surface, guiding it through the surrounding sea with gentle, coordinated strokes. The larva feeds in the conventional manner, drawing in tiny particles and digesting them internally. Its digestive tract functions simply and efficiently, converting suspended organic matter into the energy required for growth and motion. In these early days, it resembles countless other marine larvae, indistinguishable to the untrained eye, part of the vast drifting community that inhabits the water column. Yet, within its tissues lies a different destiny. As it moves, it responds to faint chemical gradients that thread through the water. Dissolved compounds, subtle traces rising from the seafloor, create an invisible map. The larva does not see this map, but receptors along its surface register changes in concentration. Gradually, its swimming becomes directed rather than random. It descends in increments, adjusting its position as signals strengthen. The search may last days or weeks. Many larvae will never encounter the precise conditions required for transformation. Currents disperse them widely. Predators intercept some. Others exhaust their limited reserves before finding a suitable site. Survival depends on coincidence, guided by sensitivity, on drifting into the narrow envelope where chemistry aligns with inherited potential. When the larva finally reaches a patch of sediment infused with the right balance of compounds, its behavior shifts. Swimming slows. The body tilts downward. Contact with the substrate triggers a cascade of internal changes. Cilia that once propelled it through open water begin to diminish. The larva secretes the first thin layer of material that will anchor it in place. This moment of settlement is both quiet and irreversible. The organism transitions from motion to permanence. Its mouth, once essential, will no longer define its future. The digestive tract that sustained its early wandering will soon become unnecessary, but for now, it remains intact, carrying out its final tasks as an independent feeder. Embedded in the sediment, the young worm begins to reorganize. Cells proliferate in new patterns. Tissues thicken where protection will be needed. The drifting stage closes without ceremony, replaced by a life oriented vertically rather than horizontally, toward exchange rather than pursuit. In this shift from free swimmer to anchored juvenile, the blueprint of a 2-century existence begins to unfold, rooted in a place chosen by chemistry and chance. Change begins at a scale too small to witness directly. Within the tissues of the newly settled worm, cells divide and rearrange, following instructions long encoded in its genome. The young body, still equipped with a functioning mouth and gut, stands at a threshold between two modes of existence. From the surrounding sediment, bacteria enter through the developing tissues. They are not random intruders, but specific lineages adapted to oxidize the very chemicals rising through the seafloor. The juvenile worm absorbs them from its environment, drawing them inward through epithelial surfaces that are still permeable and flexible. Once inside, the bacteria are enveloped by host cells, housed within membrane-bound compartments where they can persist without triggering immune rejection. As these microbial partners multiply, the worm's internal architecture begins to reorganize. The digestive tract, once a simple tube running the length of the body, loses prominence. Cells lining the gut reduce in number. The lumen narrows. Nutrient absorption through feeding becomes less central as bacterial metabolism increases. Organic carbon, produced within symbiotic cells, begins to circulate through the worm's bloodstream. Gradually, the mouth ceases to function. Its opening constricts as surrounding tissues proliferate and seal it shut. Muscles that once coordinated ingestion atrophy. The gut continues to shrink, no longer required to process external food. Over time, it disappears almost entirely, leaving only vestigial traces embedded within other tissues. The transformation does not occur in a single moment, but through a steady sequence of cellular decisions, each reinforcing the next. In place of the digestive system, a new organ expands. This structure, known as the trophosome, develops from modified tissue in the midbody region. It becomes densely packed with bacteriocytes, specialized host cells that contain thousands of symbiotic bacteria each. Under microscopic examination, the trophosome appears granular and richly vascularized, threaded with blood vessels that deliver oxygen and sulfide to the resident microbes. Within this organ, chemosynthesis proceeds continuously. The bacteria oxidize hydrogen sulfide, using the released energy to fix carbon dioxide into organic molecules. A portion of these compounds is transferred directly to the host, sustaining its metabolism. The worm, in turn, supplies the bacteria with a stable environment and a steady flow of raw materials gathered through its circulatory system. This reconfiguration marks a permanent biological shift. The worm relinquishes independence in exchange for integration. It no longer consumes particles from the environment. Instead, it cultivates an internal community whose productivity replaces digestion entirely. The boundaries between host and symbiont blur, forming a composite organism shaped by cooperation at the cellular level. What began as a drifting larva with a simple gut becomes an adult whose survival depends on an internal garden of microbes. The loss of a mouth is not deprivation but specialization, a narrowing of possibility that opens into endurance measured not in days or seasons, but in centuries. Deep within the worm's elongated body, beyond the protective tube and beneath the red crown that gathers dissolved gases, there exists an organ unlike any found in animals of the sunlit world. It occupies much of the central interior, dense and softly granular, threaded with an intricate network of blood vessels. This is the trophosome, a structure grown not for digestion in the traditional sense, but for cultivation. Its tissue is composed largely of bacteriocytes, host cells swollen with symbiotic life. Each of these cells contains thousands of bacteria, and across the entire organ their numbers rise into the billions. They are not scattered loosely, but arranged in organized clusters, held within membranes that both separate and connect them to the worm's own cytoplasm. The boundary between host and symbiont is intimate, measured in micrometers. Blood flows steadily through adjacent vessels, carrying oxygen and hydrogen sulfide absorbed from the surrounding water. Within the trophosome, these compounds meet the bacteria that rely on them. Using specialized enzymes, the microbes oxidize hydrogen sulfide, stripping electrons from the molecule and passing them along biochemical pathways that generate usable energy. Oxygen serves as the final electron acceptor, completing the reaction and allowing the cycle to continue without interruption. The energy released in this process powers carbon fixation. Carbon dioxide dissolved in the bloodstream is drawn into bacterial cells and incorporated into organic compounds through a sequence of reactions comparable in precision to those that occur in green plants. Though driven by chemistry rather than light, sugars and other carbon-rich molecules are synthesized within the bacteria, forming the basis of nourishment. A portion of these newly formed compounds remains within the microbes, sustaining their own growth and division. Another portion diffuses outward into the surrounding host tissue, entering the worm's circulatory system. In this way, organic carbon flows from chemical reaction to animal metabolism without ever passing through a mouth. The trophosome functions as a living internal ecosystem. The worm regulates the supply of oxygen and sulfide through adjustments in blood flow, maintaining conditions that favor balanced microbial productivity. If sulfide concentrations fluctuate, transport rates shift accordingly. The bacteria respond to these changes by modulating their metabolic activity, increasing or decreasing carbon fixation in subtle coordination with their host's needs. Cells within the trophosome also undergo turnover. As bacteria age or become damaged, host mechanisms digest and recycle them, reclaiming nutrients and preventing uncontrolled proliferation. The garden is not static but continuously pruned and renewed, its composition shaped by feedback between microbial chemistry and animal physiology. Within this enclosed landscape, Matter is transformed quietly and without spectacle. Inorganic molecules rising from deep geological processes are converted into the substance of living tissue. The worm's longevity rests upon this sustained exchange, an internal field of microbial labor that replaces the act of feeding with a steady, unseen harvest. At the boundary where the worm meets the surrounding water, exchange unfolds in delicate filaments. The plume extends outward in soft branching structures, each one lined with epithelium only a few cells thick. Within these filaments, blood moves through narrow vessels, positioned close to the surface, separated from the sea by a membrane so thin that dissolved molecules pass across it with ease. Hydrogen sulfide drifts through the water in low concentrations, invisible yet potent. As it encounters the plume, it diffuses inward along gradients of concentration. Oxygen follows a similar path, moving from the surrounding seawater into the bloodstream. The two molecules, so often incompatible within animal tissues, enter together here, guided by a system designed to prevent conflict. Circulating through the plume is a specialized form of hemoglobin. Like the hemoglobin found in vertebrates, it binds oxygen with reversible precision, allowing the worm to gather and transport this gas efficiently, even in environments where concentrations fluctuate. Yet, this hemoglobin carries an additional capacity. Its molecular structure includes binding sites capable of holding hydrogen sulfide without allowing it to interfere with oxygen transport. In most animals, sulfide would inhibit cytochrome C oxidase, a key enzyme in cellular respiration, effectively blocking the use of oxygen within mitochondria. In the worm, hemoglobin acts as a buffer. By sequestering sulfide within its structure, it prevents the molecule from diffusing freely into tissues where it could cause harm. The sulfide remains bound during circulation, carried safely through the vascular network toward the trophosome. Blood returning from the trophosome carries carbon dioxide produced during microbial metabolism. This gas diffuses into the bloodstream and is transported back toward the plume. There, it moves outward across the same thin membranes, passing from blood into seawater along its concentration gradient. The plume thus serves as both intake and outlet, maintaining a continuous flow of essential gases. The coordination required for this exchange is subtle. Blood flow rates adjust in response to environmental conditions, increasing when oxygen is scarce or when sulfide levels shift. The worm's circulatory system, lacking a centralized heart in the vertebrate sense, relies on rhythmic contractions of vessels and body wall muscles to maintain steady movement. Within this fluid network, hemoglobin molecules cycle repeatedly between binding and release, their conformations shifting as they load and unload cargo. Through this mechanism, the plume becomes more than an external ornament. It functions as a chemical lung, adapted not only for oxygen, but for compounds lethal elsewhere. The red filaments remain exposed, balance between vulnerability and necessity, drawing in the ingredients required for an internal partnership that depends on both breath and chemistry intertwined. Longevity in this animal is not achieved alone. It depends on a relationship maintained with remarkable precision, sustained not for a season but for spans that exceed human lifetimes. Within the trophosome, billions of bacteria lie enclosed by host cells, yet they do not overwhelm their surroundings. Their growth is steady, regulated, and woven into the fabric of the worm's own physiology. The balance begins with limitation. The bacteria rely on a controlled supply of hydrogen, sulfide, and oxygen delivered through the bloodstream. These compounds do not arrive in excess. The worm's circulatory adjustments ensure that concentrations remain within ranges that favor productivity without encouraging unchecked proliferation. When environmental levels shift, internal transport shifts as well, narrowing or widening the flow like a careful valve. Within bacteriocytes, host membranes enclose each microbial population. Nutrients and waste products pass across these membranes through selective transport proteins, allowing exchange without surrendering control. The worm provides carbon dioxide, sulfide, and oxygen. In return, the bacteria synthesize organic molecules that diffuse outward to nourish host tissues. The flow of matter moves in both directions, but it is never without boundary. Cells within the trophosome are not permanent fixtures. As bacteria divide and age, host mechanisms recycle portions of the microbial community. Lysosomal pathways digest senescent symbionts, reclaiming amino acids and other components for reuse. In this way, the worm prunes its internal garden, preventing accumulation beyond what its tissues can support. Bacterial density remains high, yet it remains ordered. The microbes themselves are adapted to this enclosed existence. Over evolutionary time, many genes unnecessary for life outside the host have been reduced or lost. Freed from the need to compete in open sediment, they specialize in chemical conversion within a stable cellular environment. Their metabolic pathways align closely with the worm's needs, minimizing waste and maximizing usable output. Efficiency arises from proximity. Because the bacteria reside within the worm's body, energy-rich compounds do not need to travel across complex digestive systems or through external food webs. Carbon fixed in one cell can enter host circulation almost immediately. Little is lost to scavengers. Little dissipates into the surrounding sea. The partnership reduces the energetic cost of feeding to the regulation of internal chemistry. Over decades, this equilibrium persists. The worm does not expend energy hunting or migrating. The bacteria do not invest in structures for survival outside their host. Each benefits from the other's stability. Environmental fluctuations are buffered by physiological control, and cellular turnover prevents imbalance from taking hold. Such endurance depends on cooperation, refined at molecular scales. What appears outwardly as stillness is sustained by continuous negotiation between host and symbiont, a dialogue of metabolites and membranes carried on quietly for centuries. Time leaves its record not in rings of wood or layers of shell, but in the gradual extension of a tube. Each year, the worm adds a narrow increment to its protective casing, secreting fresh material at the margin where body meets opening. The increase is subtle, almost imperceptible in a single season, yet across decades it becomes measurable. Marine biologists studying these animals cannot count annual bands as they might in trees or clams. Instead, they estimate age by combining tube length with observed growth rates. Long-term monitoring of marked individuals, along with radiometric dating of surrounding carbonates, has revealed a striking pattern. Many specimens lengthen by only a few millimeters per year. Some extend less than 1 centimeter over an entire decade. When a tube reaches lengths exceeding 2 meters, simple arithmetic begins to suggest extraordinary lifespans. Even accounting for faster growth during early years and slower extension later, the models converge on ages measured in centuries. The tube becomes a quiet chronicle, recording persistence rather than speed. This restrained growth is not merely a consequence of environmental limitation, it reflects a physiological strategy. The worm's metabolism operates at a reduced rate compared to shallow water relatives. Oxygen consumption is modest. Cellular turnover proceeds slowly. Energy derived from internal symbionts is allocated toward maintenance and incremental expansion rather than rapid reproduction or movement. In many organisms, High metabolic rates correlate with shorter lifespans. Rapid energy use can lead to increased production of reactive oxygen species, molecules capable of damaging DNA, proteins, and membranes. Over time, accumulated damage contributes to aging. In the deep-sea worm, slower metabolic flux appears to moderate such effects. Fewer free radicals are generated per unit time, and repair processes keep pace with wear. Cold temperatures further temper biochemical reactions. Enzymatic processes proceed steadily but without haste. The surrounding pressure stabilizes proteins and cellular structures, reducing mechanical stress. Within this context, growth becomes a measured act, aligned with an environment that changes little from year to year. The worm does not rush toward maturity. Its reproductive output is periodic and restrained, conserving resources for continued survival. Tube extension continues throughout life, yet always at a controlled pace. Each additional millimeter represents energy carefully budgeted and converted into structure. Seen across a human lifetime, the difference in length may appear negligible. Seen across 2 centuries, it becomes substantial. The animal's unusual pace forms the foundation of its longevity. By living slowly, by converting chemical energy into tissue with deliberate economy, it stretches existence across spans that outlast shifting histories at the surface, marking time not in seasons but in slender increments of growth. Numbers emerge slowly from the deep, assembled not from a single measurement, but from overlapping lines of evidence. No calendar marks the beginning of a worm's life. No direct record accompanies its first settlement into sediment. Age must be inferred through patient comparison, through models that combine observation with chemistry. One approach begins with growth. By tracking individuals over years, researchers establish average extension rates for the protective tube. Remotely operated vehicles return to the same clusters, measuring length changes with calibrated lasers and imaging systems. Early growth appears somewhat faster, then tapers into a remarkably steady pace. When these rates are applied to the full length of mature specimens, projected ages extend far beyond a human lifetime. Growth alone, however, is not sufficient. Conditions may vary subtly across sites, influencing how quickly an individual elongates. To refine estimates, scientists examine mineral deposits associated with the worms. Carbonates precipitate from seep fluids and accumulate around tubes, incorporating carbon isotopes whose ratios can be analyzed. Radiometric dating of these deposits provides timeframes for when particular structures formed. When tube sections are embedded within carbonate layers of known age, a chronological anchor becomes possible. These methods do not yield exact birthdays. They produce ranges, constrained by known rates and measured isotopic decay. Statistical models incorporate uncertainty in growth variability, environmental change, and sampling resolution. The result is not a single number, but a probability distribution. Within that distribution, ages exceeding 200 years recur consistently for the largest individuals. Margin of error remains part of the calculation. A worm estimated at 220 years might in reality be somewhat younger or older. Yet, even when conservative assumptions are applied, trimming the upper bounds of growth projections and accounting for potential acceleration in early life, the figures rarely fall below a century and a half. The convergence of independent approaches strengthens confidence in the conclusion. Comparisons with related species also inform interpretation. Hydrothermal vent tubeworms, living in more volatile settings, grow rapidly and reach maturity within years. Their shorter tubes and documented lifespans provide a contrasting baseline. In the stable seep environment, the same anatomical framework unfolds over far longer intervals. The disparity in growth rates aligns with environmental constancy and reduced metabolic demand. Over time, the accumulation of measurements has transformed initial speculation into cautious consensus. When multiple lines of evidence point towards similar durations, the idea of a 200-year life ceases to be extraordinary and becomes simply descriptive. The worm's slow extension, set against isotopic clocks embedded in surrounding minerals, composes a timeline measured in centuries, grounded in data drawn carefully from the deep. Beneath the seafloor, far below the living clusters that rise into dark water, vast stores of carbon lie compressed within layers of sediment. These deposits formed over millions of years as microscopic plankton settled, were buried, and slowly transformed under heat and pressure. The result is methane held within porous rock, trapped in pockets, or locked into crystalline structures known as gas hydrates. These reservoirs do not empty quickly. They release their contents gradually, through fractures and permeable pathways that open and close over geological time. Cold seeps draw from these deep supplies. As long as pressure conditions remain stable and subsurface channels persist, methane continues to migrate upward. The movement is measured, often slow enough that flow rates appear constant across decades of observation. Hydrogen sulfide, generated by microbial activity within sediments that consume methane anaerobically, accompanies this upward drift. Together, these compounds sustain the chemical gradients that define the seep. Unlike hydrothermal systems driven by magmatic heat, cold seeps rely on sedimentary processes that shift gently rather than violently. There are no eruptions, no sudden bursts of superheated fluid. The temperature of the surrounding water remains nearly unchanged. Geological structures beneath the site may evolve, yet they do so at a pace aligned with tectonic drift rather than seasonal fluctuation. A seep can persist in roughly the same location for hundreds or even thousands of years. This constancy shapes the biological community anchored above it. Organisms adapted to the chemical conditions experience minimal environmental variability. Storms at the surface do not disturb the deep substrate. Currents at depth move slowly and predictably. Chemical flux may rise or fall slightly, but rarely does it vanish without warning. For a worm whose growth is measured in millimeters per year, such reliability provides the foundation for extended life. Environmental stability reduces the need for rapid reproduction or dispersal. Energy can be invested in maintenance rather than migration. The internal partnership with bacteria benefits from consistent sulfide availability, allowing metabolic processes to operate within narrow, optimal ranges. When external conditions fluctuate only subtly, physiological systems avoid repeated stress responses that would otherwise consume resources. Geological methane reservoirs act as slow-burning archives of ancient carbon, feeding the seep long after surface climates have shifted and coastlines have redrawn. As long as sediment layers remain intact and pressure balances are maintained, the upward seepage continues. In this enduring exchange between rock and water, the stage remains set for organisms that depend on chemical constancy. Longevity here is not an isolated trait, but an outcome of context. The worm's extended lifespan reflects the measured rhythm of the earth beneath it, a rhythm governed by reservoirs laid down in distant epochs and released with patience that spans centuries. Stillness does not mean safety. Even in this chemically sustained landscape, movement threads between the anchored forms. Shadows pass over pale tubes, subtle shifts in the surrounding water revealing the presence of other inhabitants that share the seep. Among the most persistent visitors are crabs adapted to life at depth. Their legs move with measured precision across carbonate crust and sediment, claws probing crevices and testing exposed surfaces. Some species graze on microbial films, while others investigate the soft tissues of animals that extend beyond protective coverings. A plume left retracted too slowly can become vulnerable to a swift pinch. The red filaments, rich with blood and nutrients, offer concentrated energy to an opportunistic predator. Fish also patrol these communities. Eelpouts and other bottom-dwelling species weave through clusters of shells and tubes, guided more by chemical cues than by sight. Their movements are unhurried, conserving energy in cold water, yet attentive to any sign of weakness. A damaged tube, a partially exposed body, or a juvenile not yet fully anchored can attract attention. Despite these threats, adult tubeworms are rarely consumed whole. The length and rigidity of their tubes provide a significant defense. Buried deeply in sediment at the base and extending upward well beyond the reach of many benthic predators, the tube acts as both barrier and refuge. When disturbed, the worm can withdraw rapidly, sliding its soft body downward into the casing until only the sealed opening remains. The material of the tube resists casual breakage. Crabs may scrape at its surface, but penetrating to the living tissue inside requires sustained effort. Over years, the tube thickens incrementally, adding layers that increase durability. Its vertical orientation also reduces the area accessible to ground-based predators, presenting a narrow profile rather than a broad target. Juveniles face greater risk. Their tubes are shorter, their withdrawal distance limited. Mortality in early stages likely shapes population structure, with only a fraction of settlers surviving long enough to achieve substantial length. Yet once a worm reaches maturity and its tube extends well above the sediment, predation pressure diminishes. Predators at seeps do not eliminate the tube worms. Instead, they participate in a balanced exchange, removing damaged individuals, feeding on exposed tissue, and integrating the worms into a broader food web. The seep community remains dynamic, not a static garden, but a system of interactions moderated by structure and adaptation. In this environment, defense is architectural rather than aggressive. The worm does not fight or flee across the seafloor. It builds upward, encasing itself in a gradually lengthening tower that turns vulnerability into distance. Over decades, that distance becomes protection, allowing a soft-bodied animal to persist amid the quiet patrols of the deep. In most animals, survival begins with pursuit. Muscles contract, senses scan, bodies move through water or across land in search of nourishment. Here, that pattern has been set aside. The worm stands anchored, its body enclosed, its tissues sustained without any act of capture or consumption in the familiar sense. Energy reaches it from within. Through the plume and circulatory system, dissolved compounds enter the bloodstream and are carried inward to the trophosome. There, symbiotic bacteria transform hydrogen sulfide and carbon dioxide into organic molecules that diffuse directly into host tissues. Sugars, amino acids, and other metabolites appear not as the result of chewing or swallowing, but as products of internal chemistry. The worm's cells receive these compounds already synthesized, ready for incorporation into growth and maintenance. Without a mouth, there is no digestive tract to maintain. No gut lining must be renewed after abrasion by food particles. No enzymes must be secreted into a lumen and later broken down. The energetic cost of peristalsis, of processing bulk material, of expelling indigestible waste, is absent. Metabolism becomes streamlined, focused on distribution rather than breakdown. Hunting, too, requires expenditure. Movement through dense water demands muscular effort. Sensory systems that track prey must be supported by neural tissue, itself metabolically expensive. In relinquishing mobility and external feeding, the worm avoids these costs. It does not chase, stalk, or compete in bursts of speed. Its position remains fixed, and its energy budget reflects that stillness. The partnership with bacteria transforms the environment into a constant supplier. As long as chemical gradients persist, substrates for chemosynthesis arrive continuously. There is no feast and famine cycle dictated by drifting organic matter. Instead, nutrients are produced steadily at the site of use. The trophosome functions as a controlled reactor, matching output to demand through regulation of blood flow and symbiont density. This internal delivery system enhances efficiency. Organic carbon generated within bacteriocytes moves only short distances before entering host metabolism. Losses to the surrounding environment are minimal. The conversion pathway from inorganic molecule to worm tissue is direct, bypassing intermediate trophic levels. Stillness, in this context, is not passivity but design. By eliminating the need for a mouth and the behaviors associated with feeding, the worm reduces variability in its energy intake. Its survival depends less on chance encounters and more on the reliability of chemical supply. Over decades and centuries, this efficiency accumulates into endurance. Where many animals must continually seek sustenance, this one cultivates it internally. In the absence of jaws and gut, it finds stability, allowing its life to unfold without pursuit, anchored in a system that delivers nourishment quietly from within. In another region of the deep ocean, far from the slow exhalations of cold seeps, a different tubeworm rises from the seafloor. Riftia pachyptilla inhabits hydrothermal vents along mid-ocean ridges, where seawater percolates into the crust, becomes superheated, and returns laden with minerals. The setting is volatile, marked by steep temperature gradients and episodic bursts of fluid. Here, chemical energy surges upward in pulses rather than steady streams. Riftia shares the same fundamental strategy of internal symbiosis. It lacks a mouth as an adult, houses chemosynthetic bacteria within a trophosome, and extends a red plume into surrounding water. Yet the tempo of its life differs profoundly. Growth rates measured at vents reveal astonishing speed. Juveniles can reach lengths of over a meter within a few years. Tubes elongate rapidly, supported by abundant sulfide and high metabolic throughput. The vent environment provides rich chemical supply but little stability. Chimneys collapse. Fluid pathways shift. Temperatures can fluctuate dramatically across short distances. In response, Riftia invests in rapid development and early reproduction. Its tissues are built for swift expansion, its metabolic rate comparatively high. Oxygen and sulfide are delivered in concentrated flows, fueling accelerated carbon fixation within its symbionts. This intensity carries trade-offs. The same elevated metabolism that drives fast growth also increases cellular turnover and biochemical stress. Reactive byproducts accumulate more quickly. Structural proteins and membranes experience greater flux. Lifespans measured in Riftia are counted in decades at most, often far less. The environment itself may not persist long enough to support centuries of occupancy. By contrast, Lamellibrachia inhabits seep systems where chemical input is modest but enduring. Growth unfolds gradually, millimeter by millimeter, supported by lower sulfide concentrations and cooler, more uniform temperatures. Metabolic rates remain subdued. Energy allocation favors maintenance and long-term stability rather than rapid expansion. The difference in lifespan reflects this divergence in pace and context. Where Riftia thrives in abundance that may vanish within years, Lamellibrachia endures in constancy that can last centuries. One species embodies responsiveness to dynamic flux, the other alignment with geological patience. Both worms rely on the same underlying partnership with bacteria, yet their histories diverge according to the rhythms of their habitats. In the vent fields, life surges and recedes with tectonic breath. At seeps, it settles into continuity. Lamellibrachia's extended longevity arises not from a fundamentally different biology, but from a calibration of that biology to an environment that changes slowly, allowing a body built for stillness to persist long after faster-growing relatives have disappeared. Two kinds of chemical landscapes exist in the deep ocean, each shaped by forces rising from beneath the crust. One unfolds slowly, almost imperceptibly, as hydrocarbons migrate through sediment and diffuse into surrounding water. The other erupts from fissures in newly formed seafloor, where seawater heated by magma returns in mineral-rich plumes. Both sustain life without sunlight, yet their character differs in tempo and intensity. Cold seep ecosystems are built upon gradual release. Methane and hydrogen sulfide seep through porous layers, creating stable gradients that persist across long intervals. Temperatures remain close to the ambient deep sea, and chemical concentrations fluctuate within narrow bounds. Organisms inhabiting these sites experience continuity rather than abrupt change. Tubeworms, clams, and mussels anchor into sediment and grow at measured rates, their symbiotic bacteria drawing steady energy from consistent supplies. Hydrothermal vent ecosystems operate at a different scale of volatility. Fluids emerging from vent chimneys can exceed hundreds of degrees Celsius before mixing with frigid seawater. Chemical concentrations shift sharply over centimeters. Chimney structures form, collapse, and reform as tectonic forces reshape the ridge axis. The habitat itself may be transient, persisting for years or decades before waning or relocating. These environmental contrasts influence biological strategy. At vents, high sulfide concentrations and elevated temperatures support rapid metabolic throughput. Animals grow quickly, reproduce early, and exploit the abundance while it lasts. Lifespans are comparatively short, aligned with the dynamic nature of the habitat. When a vent field becomes inactive, its community must disperse or perish. At cold seeps, reduced stress allows for a different allocation of energy. Lower temperatures, moderate reaction rates. Chemical inputs, though less concentrated, endure over centuries. Organisms can invest in long-term maintenance rather than urgent expansion. Tubeworms extend their tubes incrementally, their symbiotic systems tuned to stability rather than surge. Environmental stress manifests not only in physical disturbance, but in biochemical demand. Rapid fluctuations in temperature and chemistry challenge protein, stability, and cellular regulation. In vent species, mechanisms must respond swiftly to protect against extremes. At seeps, such acute stress is rare. Physiological systems operate within narrower margins, reducing cumulative wear over time. Both ecosystems demonstrate that life can flourish in the absence of sunlight. Yet the distinction between hot and cold, between flux and steadiness, shapes the rhythm of survival. Where vents pulse with geological immediacy, seeps breathe with prolonged restraint. In that restrained setting, longevity becomes possible, grounded in an environment whose changes unfold slowly enough to accommodate lives measured not in bursts, but in centuries. Beneath hundreds of meters of water, chemistry proceeds at a measured pace. Temperature, pressure, and resource availability converge to shape the tempo of every cell. In this realm, metabolism does not surge, it hums. Cold temperatures slow molecular motion. Enzymes still catalyze reactions. Membranes still permit transport, but the rate at which substrates collide and transform is reduced. For the tubeworm, whose internal symbionts generate organic carbon through steady chemical conversion, this moderation becomes an advantage. Energy production aligns closely with demand, avoiding the peaks and troughs common in more variable environments. Within each cell, mitochondria consume oxygen and oxidize substrates derived from the trophosome. The byproducts of these reactions include reactive oxygen species, molecules capable of altering proteins and nucleic acids if not neutralized. In organisms with high metabolic throughput, such reactive compounds accumulate rapidly, requiring robust repair systems to prevent damage. In the deep sea worm, lower metabolic flux means fewer reactive molecules generated per unit time. Cellular maintenance keeps pace with wear. Pressure at depth exerts additional influence. High hydrostatic pressure compresses biomolecules, stabilizing certain protein conformations and affecting membrane fluidity. Deep sea species possess adaptations that maintain proper function under these conditions, including altered lipid compositions and pressure-tolerant enzymes. The environment, though extreme by surface standards, becomes internally normalized. Structural stability reduces the likelihood of mechanical stress disrupting cellular processes. Energy budgeting reflects these constraints. Rather than investing heavily in rapid growth or frequent reproduction, the worm channels resources into preservation. DNA repair pathways remain active. Proteostasis networks manage misfolded proteins. Cell division occurs, yet at a cadence that balances renewal with conservation. Tissues are replaced gradually, limiting the accumulation of replication errors. Cold water also influences ecological dynamics. Predation, pressure, resource pulses, and physical disturbances are muted compared to shallower zones. Without the need for bursts of escape or competition driven by sudden scarcity, physiological systems remain within steady operating ranges. Hormonal fluctuations that might accompany acute stress are less frequent, further reducing metabolic strain. Over years, this moderated pace compounds. Each day involves modest energy turnover, modest production of potentially damaging byproducts, and modest repair. The cumulative effect is longevity not through suspension of time, but through alignment with an environment that favors steadiness over intensity. Metabolism in the deep does not seek acceleration, it favors continuity. In the quiet balance between energy generation and expenditure, between molecular damage and repair, the worm's life extends outward, its internal rhythms synchronized with cold water and heavy pressure that shape existence into a long, unhurried arc. Seasons pass above in cycles of light and storm, yet far below, the passage of time carries a different texture. At the ocean floor, change is measured less in days than in decades. The water remains cold and dense, moving in broad currents that shift slowly, almost imperceptibly. Light never flickers across this landscape. The rhythm is geological. Year to year, the contours of the sediment alter only slightly. Fine particles settle from distant waters, forming thin layers that accumulate like quiet snowfall. A crab may trace a path across the substrate, leaving a temporary imprint that softens within hours. A fish may glide between structures, stirring a faint cloud that soon resettles. These movements are small and localized, unable to transform the broader scene. Storms that churn the surface lose their force long before reaching such depths. Waves expend their energy in shallower layers. Even powerful hurricanes, dramatic above, translate into muted pulses below. Pressure remains constant, temperature steady, chemical gradients stable within narrow ranges. The environment does not oscillate between extremes of heat and cold, abundance and scarcity. In this subdued setting, biological processes unfold without interruption from sudden upheaval. Tubes extend incrementally, shells thicken, microbial communities adjust to minor fluctuations in chemistry, but are rarely forced into abrupt shifts. Disturbance events occur, yet they are infrequent and often localized, tied to slow tectonic movements or gradual shifts in seep activity rather than rapid external forces. Deep sea stillness is not absence of motion, but reduction of variability. Currents flow, yet they do so with consistency. Organisms interact, yet their exchanges are deliberate. The pace of decay is slower, the pace of growth restrained. Without cycles of intense sunlight or seasonal blooms, the background against which life proceeds remains largely unchanged. This continuity shapes perception of time. A worm that lives for 2 centuries experiences a world that, from its perspective, alters very little. The sediment at its base may thicken by a few centimeters. Nearby structures may shift subtly as carbonates accumulate. Yet the overarching framework persists. In such an environment, longevity does not require resistance to constant disruption. It requires alignment with steadiness. The absence of frequent disturbance reduces the need for rapid adaptation. Energy can be directed toward maintenance rather than recovery. Here, time feels expansive not because events are dramatic, but because they are sparse. The ocean floor holds its quiet form across years, offering a stage where life can continue with minimal interruption, extending forward in the same calm configuration that existed decades before and will remain decades after. On the surface of the planet, nearly every green landscape begins with light. Photons strike chlorophyll, electrons are excited, and carbon dioxide is woven into sugars that ripple outward through food webs. Photosynthesis shapes forests, grasslands, and reefs, anchoring ecosystems to the daily arc of the sun. For a long time, it seemed inseparable from the existence of complex life. Far below, another pathway unfolds without a single ray. Chemosynthesis relies not on light, but on the rearrangement of chemical bonds. In cold seep environments, bacteria extract energy by oxidizing reduced compounds such as hydrogen sulfide or methane. Electrons flow through enzymatic chains, generating the energy needed to fix carbon dioxide into organic molecules. The end product, in fundamental terms, resembles what plants produce at the surface. Carbon becomes biomass, capable of supporting tissues, growth, and reproduction. The difference lies in the energy source. Photosynthesis captures energy that has traveled millions of kilometers from the sun. Chemosynthesis draws from gradients established within the Earth itself, from molecules shaped by geological processes. One pathway depends on the turning of the planet toward light, the other on the slow circulation of chemicals through rock and sediment. In both cases, the transformation of inorganic carbon into organic form creates the foundation for higher levels of organization. Animals that host chemosynthetic bacteria do not require leaves or plankton drifting from above. They are sustained by microbial partners that perform carbon fixation internally or in surrounding sediments. Organic matter generated in darkness can accumulate, circulate, and nourish a diverse assemblage of species. Complexity does not demand sunlight so much as it demands energy flux, whether that flux arrives as photons or as chemical disequilibrium. It can be harnessed to build cells, tissues, and communities. The structures differ in scale and tempo, yet the principle remains constant. Energy is captured, carbon is fixed, and life expands outward from that initial conversion. Photosynthesis supports the vast majority of Earth's biosphere, yet it is not the sole architect of biological possibility. In places where light never penetrates, organisms have evolved systems that tap into alternative sources, demonstrating that metabolism can be decoupled from illumination. The presence of intricate communities at seeps and vents confirms that multicellular life can persist where the sun's influence does not reach. Here, in sustained darkness, chemical energy replaces sunlight without diminishing the capacity for structure and longevity. The absence of light does not preclude complexity. It simply redirects the origin of nourishment from the sky above to the chemistry below. Far from the reach of sunlight, oxygen still finds its way into the deep. It begins at the surface, where wind and waves mix the upper ocean and allow atmospheric gases to dissolve into seawater. In polar regions, cold temperatures increase the density of this oxygen-rich water. It sinks and joins vast currents that slide along the ocean basins, traveling slowly across continents of darkness. This deep ocean circulation unfolds over decades and centuries. Water masses formed near the poles descend and spread outward, carrying with them the chemical imprint of their origin. As they move through abyssal plains and along continental margins, they retain dissolved oxygen, though concentrations diminish gradually as organisms consume it. Cold seeps lie within this circulating system. Although chemical compounds rise from beneath the seafloor, oxygen arrives from above through these slow-moving currents. It diffuses into the bottom water that bathes seep communities, maintaining levels sufficient to support aerobic metabolism. The balance is delicate. Too little oxygen would constrain both the worm and its symbiotic bacteria. Too much sulfide without adequate oxygen would disrupt the reactions that power carbon fixation. Within the plume of the worm, oxygen is absorbed and transported inward, meeting hydrogen sulfide in controlled proportions. The bacteria housed in the trophosome rely on oxygen as the final electron acceptor in their metabolic pathways. Without it, the oxidation of sulfide would stall, and the generation of energy required for carbon fixation would cease. Oxygen thus becomes an essential partner in a process that otherwise unfolds in darkness. Deep waters are not uniformly oxygenated. In certain regions of the world, ocean oxygen minimum zones develop where circulation is weak and biological consumption high. Cold seep communities tend to occur where circulation maintains moderate oxygen availability. The slow but persistent renewal of deep water ensures that levels do not fall below thresholds required for aerobic life. Oxygen also influences broader community structure. Fish and crustaceans that move among seep organisms depend on it for their own respiration. Microbial processes within surrounding sediments shift according to its presence or absence, shaping the chemical gradients that define the habitat. The journey of oxygen from surface to depth is gradual and largely invisible. It does not arrive in bursts, but in steady supply, replenished as currents circulate around the globe. In this way, even in places untouched by light, a connection remains to the atmosphere above. The worm's longevity rests partly on this continuity. Chemical energy rising from the earth meets oxygen descending from the sky, and at their intersection, life persists in measured equilibrium. sustained by currents that link distant regions into a single, circulating whole. Within, the worm's circulating blood moves a molecule that carries more than breath. Suspended in plasma at high concentration, hemoglobin gives the plume its deep crimson color, yet its function extends beyond the familiar task of oxygen transport. In this animal, hemoglobin has been shaped by evolutionary pressure to manage a compound lethal to most life. Hydrogen sulfide interferes with cellular respiration by binding to enzymes in the mitochondrial electron transport chain. In typical animals, even low concentrations can halt energy production, disrupting the delicate chemistry that sustains cells. At cold seeps, sulfide is not a trace contaminant, but a defining feature of the habitat. For an organism dependent on sulfide oxidizing symbionts, exposure is unavoidable. The worm's hemoglobin resolves this tension through structural modification. Its protein subunits contain binding sites capable of reversibly attaching hydrogen sulfide without compromising the molecule's affinity for oxygen. Sulfide and oxygen are carried simultaneously, held in distinct chemical contexts within the same circulating complex. This arrangement prevents free sulfide from diffusing into sensitive tissues while ensuring its delivery to the trophosome. As blood flows from the plume inward, hemoglobin binds oxygen absorbed from surrounding water. At the same time, sulfide molecules attach to specific regions of the protein, shielded from reactive interaction with other cellular components. Transport occurs through an intricate vascular network that distributes these gases precisely where they are required. When the blood reaches the trophosome, local conditions favor release. Bacterial symbionts utilize sulfide as an energy source, oxidizing it in controlled reactions that power carbon fixation. Oxygen, delivered in parallel, serves as the terminal electron acceptor in this metabolic sequence. Hemoglobin thus acts as both courier and safeguard, ensuring that substrates arrive intact and in balance. The biochemical adaptation extends to regulation. Variations in environmental concentration prompt subtle shifts in binding dynamics. The worm can modulate blood flow and plume exposure, influencing how much sulfide and oxygen enter circulation. Hemoglobin's capacity to accommodate both molecules allows flexibility without toxicity. This molecular refinement did not arise instantly. Over generations, natural selection favored protein variants capable of tolerating and managing sulfide-rich conditions. Amino acid substitutions altered binding pockets, refining specificity and stability. The resulting hemoglobin differs in structure from that of surface-dwelling animals, yet it remains recognizable as the same fundamental protein. Through this adaptation, a potential poison becomes a resource. Tissues remain protected, respiration proceeds uninterrupted, and symbiotic metabolism thrives. The worm's longevity depends in part on this quiet biochemical negotiation carried out by hemoglobin molecules that circulate continuously, maintaining equilibrium between danger and necessity in the chemistry of the deep. Around the anchored tubes, life gathers in quiet layers. The seep is not a solitary stage, occupied by a single species, but a mosaic of organisms linked by shared chemistry. Each participant draws from the same underlying gradients, yet each does so in a distinct way. Mussels cluster in dense beds along carbonate outcrops and sediment margins. Their shells overlap, forming living mosaics that filter surrounding water. Many harbor their own symbiotic bacteria within gill tissues, converting methane or sulfide into organic carbon, much as the worm's internal partners do. Valves open and close slowly, regulating exposure to dissolved compounds while maintaining a steady exchange with the environment. Clams embed themselves more deeply in the sediment. Their siphons extend upward, drawing water downward through buried bodies. Within specialized tissues, symbiotic microbes fix carbon, supplying nourishment directly to their hosts. The shells thicken over time, layering calcium carbonate in response to chemical conditions that persist year after year. Some individuals may live for decades, contributing to the structural complexity of the site. Shrimp move more freely among these anchored forms. They traverse the substrate with deliberate steps, antennae sampling chemical cues. Some graze on microbial films, coating hard surfaces. Others scavenge organic matter that settles between tubes and shells. Their mobility introduces a subtle circulation of nutrients, connecting patches of productivity within the seep. Across the sediment itself, microbial mats spread in pale or iridescent sheets. These communities consist of bacteria and archaea, adapted to thrive at the interface where methane, sulfide, and oxygen meet. They form thin layers that ripple gently in passing currents, mediating chemical flux between sediment and water. Through their metabolism, they influence the gradients that support larger organisms, shaping the availability of substrates throughout the habitat. No single species operates in isolation. Organic compounds generated by symbionts enter surrounding waters and sediments, becoming resources for others. Waste products from one metabolic pathway serve as inputs for another. Carbon fixed in darkness moves through shells, muscles, and exoskeletons, woven into a network of exchange. Predators thread through this assemblage, removing individuals and redistributing biomass. Decomposers break down remains, returning nutrients to the system. The seep functions as a localized but intricate ecosystem, structured by chemical energy, yet diversified through biological interaction. In this interconnected community, the tubeworm is both participant and architect. Its tubes provide vertical structure, surfaces for colonization, and refuge from currents. Mussels, clams, shrimp, and microbes respond to gradients that the seep sustains, forming a layered society anchored in sediment but animated by shared metabolism. Within the stillness of the deep, complexity unfolds quietly, sustained by chemistry and bound together through exchange. Reproduction unfolds here without display, carried out in water that never brightens. The adult worm remains fixed within its tube, yet within its tissues, gametes mature in quiet cycles. Male and female individuals release sperm and eggs into the surrounding current, not through pairing or courtship, but through broadcast spawning. Timing is subtle, often synchronized by shifts in chemical cues or seasonal patterns in deep water circulation that are faint yet detectable. Once released, eggs drift outward, buoyed by currents that move steadily along the seafloor. Fertilization occurs externally, and the resulting embryos develop into free-swimming larvae. For a brief span, they inhabit a different existence than their anchored parents. Cilia propel them through the water column, allowing them to navigate chemical gradients and maintain position within favorable layers. Dispersal is both opportunity and risk. Currents may carry larvae kilometers from their origin, widening the genetic exchange between distant seep communities. This connectivity prevents isolation and maintains diversity within populations that might otherwise remain fragmented across the ocean floor. Yet the vastness of the deep sea means that many larvae never encounter suitable habitat. The chemical signatures that define a seep are localized, faint threads in an immense volume of water. As larvae drift, sensory receptors detect subtle increases in dissolved compounds associated with active seepage. Concentrations of methane, sulfide, and other byproducts form invisible trails that guide descent. The process resembles orientation toward a distant source, though no light marks the path. Gradual adjustments in swimming direction bring the larva closer to the sea floor, where conditions align with its developmental requirements. Settlement occurs only when specific thresholds are met. The larva contacts sediment, infused with the right balance of chemicals, and begins the transformation toward symbiosis described earlier. Those that settle in unsuitable areas cannot survive long without internal bacterial partners. Success depends on encountering an active seep within the limited duration of the larval stage. Over decades, new seeps may emerge as geological pathways shift while older ones wane. Larval dispersal ensures that colonization can occur when fresh habitat becomes available. In this way, reproduction extends beyond individual survival, maintaining continuity across generations and across space. The cycle repeats quietly. Adults release gametes into dark water. Larvae drift and search. A few settle and anchor. Through this restrained yet persistent strategy, populations endure despite the sparse distribution of suitable habitat. In the absence of light, reproduction relies on chemistry and current, sustaining a lineage that persists in darkness through measured release and patient dispersal. Long before any worm anchored itself in sediment, forces beneath the ocean floor were already shaping the conditions for its future. The deep seabed is not static stone, but a layered archive of shifting plates, buried carbon, and slow fracture. Over millions of years, organic matter descending from ancient seas was compressed and transformed, generating methane that became trapped within sediments and crystalline structures known as methane hydrates. Methane hydrates form under specific combinations of pressure and temperature. In cold, high-pressure environments, water molecules arrange themselves into lattice frameworks that encase methane, creating ice-like solids stable beneath the seafloor. These deposits can persist for vast intervals, storing immense quantities of carbon in frozen cages. As long as pressure remains high and temperatures low, the hydrates endure. Yet the Earth's crust is always in motion. Plate tectonics exerts quiet influence, bending and stressing the seabed. Along continental margins, sediments accumulate and compact, occasionally fracturing under their own weight. Fault lines open minute pathways that allow fluids and gases to migrate upward. When pressure conditions shift or sediments warm slightly, methane hydrates may destabilize, releasing gas into surrounding pore spaces. These releases are rarely explosive in cold seep regions. Instead, methane moves gradually through permeable layers, following networks of cracks and fissures created by tectonic strain. Hydrogen sulfide arises as microbes within sediments consume methane anaerobically, producing additional chemical gradients. The seafloor above becomes a conduit, linking deep carbon reservoirs with overlying water. Cold seeps therefore represent surface expressions of processes operating far below. Their persistence depends on continued supply from geological stores and on the structural integrity of the pathways that channel gas upward. Some seeps remain active for centuries, others for millennia, until shifts in sediment load or tectonic adjustment alter the flow. The worm's lifespan, measured in centuries, is brief compared to these underlying dynamics. Its existence unfolds within a moment of geological time. Methane hydrates formed long before its ancestors evolved. Plate movements that influence seep activity operate across intervals that dwarf any single organism's life. Yet, within this vast temporal framework, stability can prevail locally. As long as tectonic forces maintain fractures and pressure conditions support gradual methane migration, chemical gradients remain consistent. The seep becomes a stable node in a much larger system of crustal motion and carbon cycling. In this way, biological time intersects with geological time. The worm grows slowly, its tube lengthening year by year, while beneath it, plates shift almost imperceptibly, and methane reservoirs persist across epochs. Life at the seep rests upon processes that began in ancient oceans and continue through the quiet mechanics of the Earth itself. Observation begins with descent. From research vessels positioned far above, cables unwind and vehicles sink slowly through layers of darkening water. Human-occupied submersibles were among the first to carry scientists down to the seafloor, their thick hulls resisting pressure, while small viewports framed a narrow window onto the landscape below. Within those confined spheres, researchers watched tubes rise from sediment, and instruments recorded temperature, chemistry, and current. Later, remotely operated vehicles expanded the reach of study. Tethered to ships by fiber optic cables, ROVs maneuver with mechanical arms and high-resolution cameras, capable of remaining submerged far longer than any human pilot. They hover beside individual worms, illuminating their plumes and measuring dimensions with paired laser beams that project fixed distances onto the scene. These lasers create scale, allowing tube length and plume spread to be quantified precisely. Sampling occurs with careful intention. Manipulator arms collect small sections of tube or surrounding carbonate, placing them into sealed containers for analysis. Sediment cores reveal chemical gradients beneath the surface. Water samples capture dissolved gases at specific depths. Each retrieval carries fragments of a system that cannot be fully transported to the surface intact. Long-term monitoring requires return. Scientists mark selected individuals with discrete tags or rely on detailed mapping of tube clusters to relocate the same specimens years later. Photographs taken at intervals allow direct comparison. Growth can be detected by measuring the extension of tubes relative to fixed landmarks or by tracking the shift of laser reference points across time. In some studies, instruments are left behind. Autonomous landers settle on the seafloor, equipped with sensors that log temperature, oxygen concentration, and chemical flux over months or years. Data accumulates silently until retrieval. These records reveal patterns invisible during brief visits, capturing fluctuations that shape metabolic balance. Laboratory analysis complements fieldwork. Isotopic signatures from carbonate deposits inform age estimates. Molecular techniques examine the genomes of symbiotic bacteria, clarifying evolutionary relationships and metabolic pathways. Protein studies characterize hemoglobin variants, linking biochemical adaptation to environmental context. Research at depth proceeds slowly, constrained by logistics and cost. Weather at the surface can delay dives. Equipment must withstand pressure and corrosion. Yet over decades, incremental measurements assemble into coherent understanding. Each expedition refines growth models, confirms longevity estimates, and maps the distribution of seep habitats. Through submersibles and ROVs, through patient monitoring and precise measurement, scientists piece together the life history of organisms that outlast individual careers. The process mirrors the worms themselves, advancing millimeter by millimeter, guided by steadiness and careful return to the same quiet places on the ocean floor. Across the world ocean, longevity appears again and again in places where light is scarce and change arrives slowly. The tubeworm is not alone in its extended span. In cold northern waters, the Greenland shark moves through dim depths at an unhurried pace, its growth so gradual that individuals can exceed 4 centuries in age. Radiocarbon dating of eye lens tissue has revealed lifetimes that began before many modern nations existed. The shark's metabolism is restrained by low temperatures, its movements deliberate, its reproductive cycle prolonged. On continental shelves and deeper basins, the ocean quahog rests within sediment, a bivalve whose shell records time in faint growth lines. Some specimens have been aged at over 500 years. Their shells thicken incrementally, millimeter by millimeter, much like the tubes of seep-dwelling worms. Within their tissues, metabolic rates remain low, oxidative damage accumulates slowly, and repair processes persist across centuries. These disparate organisms share no close ancestry, yet they converge upon similar strategies shaped by environment. Cold water reduces enzymatic speed, lowering the rate of biochemical reactions that generate cellular stress. Limited food availability discourages rapid growth and favors efficiency. Predation pressure, though present, often unfolds at subdued tempos compared to shallower ecosystems. Depth adds another layer of stability. In the deep sea, temperature varies little from season to season. Currents shift gradually. Catastrophic disturbance is rare relative to coastal zones. Organisms adapted to these conditions align their physiology with constancy rather than fluctuation. Energy budgets emphasize maintenance, tissue repair, and slow reproduction. Longevity is not universal in the ocean, yet it clusters where environmental rhythms are extended. In surface waters, rich with plankton blooms and intense competition, rapid life cycles can confer advantage. In the deep, where resources are steady but sparse, survival favors patience, growth slows, maturation delays, and lifespan lengthens. The tubeworm at the seep mirrors this pattern. Like the Greenland shark and the ocean quahog, it inhabits cold water, maintains a low metabolic rate, and invests in structural persistence rather than rapid turnover. Its chemical partnership replaces hunting, yet the underlying principle remains consistent. Reduced metabolic intensity and environmental stability correlate with extended life. Across species and habitats, the deep sea reveals a theme. When energy flows steadily and disturbance is muted, time stretches. Organisms adapt not for speed, but for endurance. In these quiet zones of the ocean, life often unfolds on scales that surpass human expectation, shaped by cold, darkness, and the measured cadence of the deep. Aging in most animals unfolds as a gradual accumulation of molecular strain. Cells generate energy through respiration, and in doing so produce reactive oxygen species that can damage proteins, lipids, and DNA. This oxidative stress is not catastrophic at first. It builds slowly, leaving small marks that, over decades, alter tissue function. In environments where metabolism runs high, these reactions occur more frequently, and repair systems must work constantly to maintain balance. At the seep, the worm's pace of life moderates this process. Its metabolic rate remains low, aligned with cold temperatures and steady chemical supply. Energy production proceeds without the surges associated with rapid movement or fluctuating food availability. Because respiration is restrained, the generation of reactive byproducts is likewise reduced. Fewer free radicals circulate within tissues, and cumulative oxidative damage accrues more gradually. Slow cell turnover further shapes longevity. In many fast-growing animals, tissues divide rapidly to replace worn structures or to support expansion. Each division carries risk of replication error, subtle mutations that can propagate over time. In the tubeworm, growth is incremental. Structural tissues persist for years with minimal replacement. Cells in stable regions of the body may divide infrequently, preserving genomic integrity across long intervals. Repair mechanisms remain active, yet they are not overwhelmed. Antioxidant enzymes neutralize reactive molecules before they interact with essential cellular components. Proteins that detect and mend DNA lesions operate within a manageable workload. Because metabolic demand remains consistent and modest, these maintenance systems sustain equilibrium rather than racing to correct constant insult. The internal partnership described earlier also contributes indirectly. By outsourcing carbon fixation to symbiotic bacteria, the worm avoids energy-intensive foraging behaviors. It does not engage in high-speed pursuit or extensive locomotion. Muscular activity remains limited, reducing metabolic spikes that might amplify oxidative stress. Energy arrives in a measured, continuous stream rather than in pulses. Over decades, the cumulative effect of these conditions becomes visible in lifespan. Reduced metabolic damage correlates with extended functional stability. Tissues retain structural coherence. Organ systems operate within narrow physiological ranges. The organism ages, yet at a tempo that reflects its environment. This relationship between energy efficiency and aging appears across taxa. When metabolic intensity declines and environmental variability narrows, cellular wear diminishes. In the deep sea, where cold and constancy prevail, life often trades speed for endurance. The tubeworm exemplifies this exchange. Its longevity is not the result of a single protective trait, but of a suite of interconnected processes that limit oxidative stress, slow cellular turnover, and preserve biological function across centuries. In the quiet arithmetic of the deep, this worm alters familiar equations. On the surface world, sunlight anchors nearly every food web, and consumption defines survival. Plants harvest light, animals consume plants or one another, and energy moves through visible chains. At the seep, that structure dissolves into something subtler. Here, life is sustained without any reliance on solar radiation. Carbon is fixed in darkness, not by leaves, but by bacteria drawing energy from chemical bonds. The worm stands as a visible extension of invisible reactions, a structure built from processes that unfold beneath sediment and within cells. Its existence affirms that biological productivity can arise wherever gradients of energy persist, whether from photons or from the slow release of reduced compounds from the earth. The absence of a mouth does not signal absence of nourishment. Instead, nourishment arrives through partnership. By housing chemosynthetic bacteria within specialized tissue, the worm relinquishes direct feeding and entrusts energy acquisition to symbionts. In return, it provides access to substrates and protection from fluctuating conditions. Neither organism alone could occupy this niche as effectively. Together, they form a composite system in which boundaries between host and microbe blur. Symbiosis, in this context, is not a peripheral adaptation, but the central strategy. It reduces the need for movement, predation, or competitive foraging. Energy arrives in a measured flow, processed internally rather than sought externally. The worm's anatomy, physiology, and life history align around this cooperative exchange. Longevity emerges not from isolation, but from integration. The lesson extends beyond this single lineage. Across ecosystems, stable relationships often underpin persistence. When partners exchange resources efficiently and environmental conditions remain consistent, biological systems can operate for extended periods with minimal disruption. At the seep, geological steadiness supports chemical supply, Chemical supply sustains microbial metabolism, and microbial metabolism maintains the worm. Each layer reinforces the next. Stability shapes tempo. In habitats where disturbance is rare and inputs fluctuate within narrow bounds, organisms may adopt slower growth, delayed reproduction, and extended lifespan. The worm exemplifies this pattern. It does not rush through seasons or respond to rapid shifts. Its life unfolds in synchrony, with processes measured in years and centuries. From darkness, a broader understanding emerges. Life does not require light in the conventional sense. Eating is not confined to ingestion. Survival can depend on collaboration rather than competition. In the stillness of the deep, these principles converge, revealing that endurance often arises from balance, efficiency, and the quiet strength of partnership sustained over time. Far below the turning of seasons and the noise of cities, there are living bodies that began their existence before smokestacks marked the horizon. While railways spread across continents and engines reshaped landscapes, these worms were already anchored in sediment, drawing sustenance from reactions hidden beneath the seabed. They have never hunted. No pursuit has quickened their pulse. No jaw has closed around prey. They have never chewed or tasted, never tested the texture of flesh or leaf. Their nourishment has always arrived through quieter means, carried in dissolved form, translated by bacterial partners into the carbon that builds tissue year after year. They have stood upright for 2 centuries, their tubes lengthening by fractions so small they would escape notice in a single dive. Around them, generations of mussels have settled and died. Shrimp have moved through brief lifespans. Currents have shifted and returned. Above, human lifetimes have unfolded in quick succession. Yet these worms remain, their bodies adjusting slowly, their internal systems maintaining equilibrium. In stillness they persist, not immobile in the sense of lifeless stone, but steady, responsive within narrow ranges. Blood continues to circulate. Cells repair minor damage. Symbionts divide and oxidize sulfide in measured rhythms. The partnership endures as long as chemical supply remains stable, each side dependent upon the other. In chemical breath, they live, exchanging oxygen and sulfide through hemoglobin adapted for this improbable balance. What would poison most organisms becomes substrate and fuel. The deep does not offer abundance in familiar forms, yet it offers constancy. Within that constancy, life has found a way to extend itself. In partnership they endure, host and microbe inseparable in function. Energy flows not through ingestion, but through collaboration. Survival is not achieved by conquest or speed, but by alignment with gradients that change little across decades. They quietly outlive generations of surface life. Children grow, age, and pass on while the same individual worm remains rooted below. Its tube bears silent witness to centuries, measured not by calendar, but by incremental growth. There is no spectacle in this longevity, no dramatic gesture, only persistence shaped by cold water, geological stability, and efficient exchange. In darkness that never lifts, life continues at its own tempo, unhurried and sustained, a reminder that endurance can arise where light never reaches, and that time, in some places, moves with profound patience.